endocytosis in the last couple of lectures, but one of the assumptions that we had continued to make all through was the fact that the receptor number is a constant. Okay, and this is assumption that we had made time and again when we are looking at the enzymes, when we are looking at all kinds of places. We had made the assumption that the total number of a certain species is a constant, be it the total amount of enzyme, be it the total number of receptors in the system. Now, in case of enzymes, this may be true, but what happens in case of receptors is that this is not necessarily true. Why is this not necessarily true is that, I think I talked about this a little bit before, is that the body needs to regulate the number of receptors. And we talked about certain diseases, and for example, I think it did talk about the pulmonary immune function. And we said that in certain cases, the body needs to engage more receptors. In certain cases, the body might need to tone down, lower the number of receptors. So the cell has a mechanism, or the body in general, but cell in particular, has a mechanism of regulating the receptor number. So which means that the assumption that the total number of receptors is a constant is not necessarily an assumption that holds at all points of time. Okay. So what we are going to do in today's lecture is that all the model that we did in the previous lecture was based on the assumption that the total amount of receptor is a constant, if you remember, and I'll just remind you one more time that one. But what we are going to do in today's lecture is how do we model the system when the total number of receptor is not a constant, okay? Or in other words, receptor itself grows and dies, okay? So there is something, there is a process in the number of receptors is involved in the process as well, because till now the number of receptors had not been involved in the process, okay? So this is this old picture that we keep harking back at, and so here essentially that the number of receptors that you have in the system, the total number of receptors in the system here was assumed to be constant in every model that we did before, but now we are not going to assume that. So let me just go back to the model we did in the last class, this one, sorry, full kinetic model. So this was the constraint that I am talking of, nRT equals nR plus nLDLR. So the total number of receptor present as free and as complex is a constant. How did it help us? We could pull this back into this equation, and therefore it became, when CLDL is a constant equals the initial concentration, then it became a first order reaction, and we could solve it. Now we don't want to make that assumption. Before we go ahead and do that modeling, there are a couple of questions that are necessary to answer. So remember, one of the points when we talked about the mutations that results in familial hypercholesterolemia, there were 3 points, right? One was the point, first point was that the dissociation rate constant may be increased. In patients with familial hypercholesterolemia, maybe you have larger dissociation constant. What is the second point? The fact that the internalization rate constant may be lower. So in patients with familial hypercholesterolemia, the internalization rate constant may be lower. And what was the third point? The third point was maybe the dissociation rate constant and the internalization constants are all right, but what is defective is the localization in the coated pits. So the process of localization in the coated pit is not good enough. So that's why we want to ask a couple of questions based on that. So it turns out that only 2/3 of the LDL receptors are present in coated pits, yet all bind to receptor. Okay, these all the ligands that bind to these receptors. Okay, so that is the question. So can the distribution? So the questions, a couple of questions we have over here is A. Can the distribution of the LDL receptors be explained in terms of diffusion limited binding to coated pit? I discussed this a little bit, but we are going to talk about this in greater detail. And second one is, what is the relationship between the observed internalization rate constant K2 and the rate of formation of the vesicle? So these are a couple of questions I want you to think about. One of them we are going to answer in the next few minutes, which is that what is the distribution in this diffusion limited binding? So if you go back, I have to go back to the picture again and again here. So here, as you see, this is the part that we are going to talk about now. So why is that? Because we have already in the previous lecture we have already discussed the modeling of this part, that is 1 written here, binding. We have already discussed the internalization process given here by 3, for example, and the questions that are asked now, that I just now asked, are related to point number 2 and point number 4. Okay. Point number 2, what is the question? That how does this, as you can see over here, that most of these receptors go and localize in the coated pits. So the question that was asked, the question that I asked or mentioned was that the coated pits are only there very little, that most of the cell membrane is not coated pits. There are only small regions in the cell membrane which consist of coated pits, but it turns out that most of the receptors go and localize in the coated pits. So what essentially happens, what is drawing these receptors towards the coated pit, either in unbound form or in bound form, depending on the class of the receptor? You follow what I am saying? So that is question number 1, and question number 2 is here, as you see, 3 to 4, that was the other question, was that is internalization and vesicle formation the same process, or in other words, is the rate constant for internalization same as the rate constant for vesicle formation? You follow the question, that whenever you have internalization, does that mean that at the same rate vesicle formation is also occurring, right? Because these are processes in series, as I told in the morning class. These are processes in series. So first you have, say for example, binding, followed by diffusion into the coated pits, then you have internalization, and finally vesicle formation. So what is the surety that internalization is equivalent to vesicle formation? So that's another question. So what does it mean to you when I say that what is a surety that internalization is not same as vesicle formation, and if internalization is not same as vesicle formation, what should you do? What do you think you should do? It's a very simple answer, but I want, it's not a complicated question, neither is the answer complicated, but what do you think you should do? So this process, what you see on the screen here, yeah, as I said, it's a very simple answer. So here we have we have taken into account step number 1, we have taken into account step number 3, 2 we are going to take into account right now. And if you have internalized the rate of vesicle formation is not the same as the rate of internalization, just add on one more step and add a rate constant 4, okay, a reversible rate constant 4 out there, and you can have your model. So what you have to now distinguish between, you have to distinguish between the complex in the invagination or in the internalized complex as opposed to complex in the vesicle. Okay. So we had here the complex in the, here for example this is the LDL-I, which is the complex in the internalized form, then you need to add one more rate constant and go to the complex in the vesicles, so LDL-V. That is easy and we can take that into account. Now we have to talk about this part, the coated pits and what really happens. So essentially what happens is what is written on the screen. So receptors diffuse on the cell surface freely till they reach the coated pit, where they bind rapidly and irreversibly to adapted proteins to the coated pit. So what happens is that these coated pits are there. So whether it's a class 1 or class 2 doesn't matter, whether the receptor is already bound to the ligand or not bound to the ligand is not important. So these receptors will freely float around on the surface of the cell till they reach the coated pit, and coated pit consists of 3 kinds of proteins. We discussed that earlier in the picture we showed. So these coated pits, these proteins are very quick binders. So they have this ability that the rate constant for binding is very, very fast. Okay. So as a result, as soon as the receptor reaches the coated pit, it immediately binds with the coated pit irreversibly and very almost instantaneously to form the receptor on the coated pit. Okay. Now my question is that so essentially if you try and understand, there are 2 processes in the system, right? One is a diffusion, a lateral diffusion that's taking place, okay, and the second one is the binding, irreversible binding to the coated pit. So what do you think, again it's a very easy question, so what do you think would be, if I ask you to model, just let me give you this question that I'm not telling you how, if I tell you then it's anticlimax. So if I just ask you to kind of guess, come up with some open answers to, this is a system, this is what is happening in the system, so how do you model it? So if I am to draw, this is the cell surface, okay, and you have the receptors floating around here like this, and you have this region, certain region which is coated pit, it's like a sticker, okay. So as if you have put some adhesive on it. So as soon as the receptor reaches there, or with the ligand, let's say, that reaches there is stuck, it cannot move any further, so it's stuck out here. Otherwise it's freely floating, but as soon as it reaches there it's stuck. So it's like as if you've spread some gum or adhesive out there and it comes in. So now so this is the system, I talked about it, and now what do you think, how do you want to model this? If I give you, it's given an open problem. So ideally, let us say, don't worry about how difficult the computation or the calculation or the solution of the model is going to be. So if I just give you this problem in the test and say that how are you going to model the system? This is a system, how are you going to model it? So what are the processes? Tell me, what are the fundamental processes? Diffusion and reaction, irreversible reaction, it's already said, and lateral diffusion. Okay, so it is not like a normal diffusion in the sense that it's a slightly guided diffusion. Okay, slightly guided diffusion. It's not a normal diffusion, but it's slightly guided diffusion. Why is it slightly guided? Because it is directed diffusion. There is it's a lateral diffusion, but still it's some kind of diffusion. So what do you think? How should you do it? I mean, you can, if you want, come to think of it, think of diffusion in a guided channel. Like you can have a guided channel, and there can be diffusion in that guided channel, right? So that's something like that. So let's talk about the basic processes first, and then we can talk about guided channels and stuff like that. What are the basic processes? Diffusion and reaction. So are they occurring in parallel or in series? Series. So we already did this problem before in this course itself. We did this problem of diffusion followed by reaction. Okay. Now this is a tricky part. This part that we are going to do. So is a slightly tricky part. Now, if I am to actually do this whole modeling of diffusion followed by reaction, it is a very complicated process. Why? Because you don't know where the cell is, where the receptors are diffusing, A, and then at what point it is reacting. There is no direct, there is no clear cut information on to us that is available to us. Okay, do you understand? How is it different from what we had done before? What we had done before is, for example, I remember we are doing this immobilized enzyme, right? So for example, there was some reaction that is going to occur on the surface, and materials were diffusing out, and the reaction is going to occur here. So what was the advantage there? You had clear geographical understanding of what area the diffusion is going to take place, which is this area, followed by where exactly the reaction is going to occur. Here the problem is that you don't have clear understanding of where the diffusion is going to occur, which direction the diffusion is going to occur, A, and B, where exactly the coated pits are. You are not aware. I can tell you that coated pits form 5% or 10% of the entire cell membrane. I think I gave you number already, which is like that. So 10% of the entire cell membrane is coated pit, but you don't know where the 10% is, right? So you don't have clear-cut information available to you as to where diffusion is going to occur and where reaction is going to occur. So what is the process in that case? If you don't have clear-cut information available, what is this kind of modeling that we had done in the enzyme and in the immobilized enzyme? What is this modeling called? What is this? There is two broad kinds of models, model types. Two very broad types of models, and we are at the end of the course, and we have done this kind of modeling also. What? Matrix? No. Two very broad kind of models. Let me write down. Deterministic model, and the second one is probabilistic or stochastic. Okay. What is the difference between a deterministic model and a stochastic model? Everything is deterministic. What did Einstein say? He said that God doesn't play dice. Okay, which means that there is nothing that is probabilistic in the universe. Everything is deterministic. But if you can determine everything down to the last, if all information is available, then you can determine everything. For example, we all know about the butterfly effect, right? That if a butterfly flaps its wings in the Amazon, then there could be thunderstorms in Australia, rains in Australia. So that's the butterfly effect. That means the cause and effect. So every little thing leads to every little thing. So can you predict deterministically this whole process of what's happening in Amazon and what's happening in Australia? Yes, you can, provided all kinds of data is available to you. What do you mean by all kinds of data? You have to know what is the ocean currents, what the fluid mechanics of the ocean is. You have to know what the fluid mechanics of the wind is, you have to know what kind of impedances are there in the system, so you have to know all kinds of things. So once all these information is available to you, then you can write a full deterministic model that can predict from the starting from the t equals 0, the butterfly flaps its wings, and maybe 24 hours later or a couple of days later there is a thunderstorm in Australia and everything could be described. But when you cannot describe that, then you resort to probabilistic methods. Okay, what I want to impress upon you, the fact is that there is nothing probabilistic, everything is deterministic, but the fact is that we do not have access to such large amounts of information, and as a result we resort to probabilistic or stochastic model, right? Is it clear? So probabilistic or stochastic model is a result of the fact that we are deficient in information. Similarly here in this example that I was showing you, so this is a deterministic model, but this model is we have to go for something like a stochastic model. The reason we have to go for something like a stochastic model is because we do not have enough information on the system. Okay, we do not have complete information that where my coated pits are, what is the diffusion pathway that a receptor will take. If I had this information, if I was monitoring a particular cell and the whole receptor ligand binding process and the migration of the coated pits to a certain region, then I would be able to come up with a deterministic model. If I were to do a deterministic model, then what I would do is I would write a diffusion model, free diffusion, followed by these certain areas. So essentially if you look at the cell membrane, if you something like this, then my receptor is, if this is my receptor, the receptor is moving around everywhere, and you have these small regions over here which are like glues. Okay, you will have these small regions. So whenever the receptor comes over here, it sticks, and then what did you model, what did you keep a count on in that case? You would keep a count on the, what would be the thing that you would model? Is it the concentration? What is it? Number of what? What receptors? Free receptors. Number of free receptors. Okay, so you are keeping a count on the NRF, let's say, or something like that. So number of free receptors. So the number of free receptors are roaming around everywhere, and as soon as they come and there is a coated pit out here, they stick to the coated pit, which means then you immediately, so that's like a sink term. Okay, so that's like a sink term in your model, and you get rid of that, you remove that. So that would be a deterministic way of looking at it. But given this, this kind of modeling would take a lot of time, and it's possible, but again it's not impossible, but it's possible, but it will take a lot of time. So given those constraints, what we come up with is a stochastic kind of model. Okay, and the stochastic kind of model is essentially based on Brownian motion, random motion theory, Brownian or kinetic or whatever. So, and I'll show you that in a minute what we do. But what we do, probably it's the first time you are encountering something like this, is that we model the entire diffusion and reaction process that happens over here as a reaction process. Okay, but is that a reaction process? No. Is it limited by the diffusion or the reaction? What do you say? Diffusion. Of course it's limited by the diffusion, which means that diffusion is a dominant process over here, but we model it as a reactive process. So one of the things I do not want you to have an impression is that when I write rate constants, these are not rate constants of the receptor binding to the coated pit. No, because those we have already discussed, those are very large numbers, large rate constants, and those are not the numbers. Okay, so this is the overall diffusion and the reaction modeled together as a diffusive process. Okay, so let's go back to the screen. So this happens, and the complex is then internalized when a vesicle is formed. Now this is, before we go into the model, this is the this is the definition that you need to have. So now the total number of receptors that are there, apart from the ones, say let's forget the ones that have been internalized, there's just the total number of receptors at the start of the process, we divide them up into two different kinds, one which are on the pits and one which are not on the pits, the rest of the cell surface. Okay, so NRT is the total number of receptors at time t equals 0, which means that nothing has been internalized yet, equals NRP, which is on the coated pits, plus NRN, which is on the cell surface. Fine. So now this is how the process looks. Okay. So here, so this is the these are the free receptors NRN, I think, no, NRN. This is the NRN, and this would be NRP now. Fine. So these receptors are moving in this direction and moving away, and this is the coated pit, and 2b is the distance between two coated pits. Okay, and we will also have the radius of the coated pit. Once the coated pit is formed, we will have the radius of the coated pit. I will come to that, but look at these two rate constants out here, on the top here, K plus and K minus. So what are these rate constants? K plus is the affinity of the receptor towards the coated pit. Okay, so it is a random diffusional event out there plus the reactive uptake or reactive binding to those two, and K minus is the lack of affinity or the tendency of the receptor to move away from the coated pit, because though it is unlikely that a receptor that is there is going to move away from the coated pit, but still we include it. Can you tell me why? So it is very unlikely, because the rate constant is very high in the forward direction, but still we include this K minus. Why is that? And it is a something to do with the very basics. Sorry, yeah. So why do we have K plus? Fine, K plus, there is no argument or no doubt about K plus. Why do we have K minus? See, though the backward rate constant is low, but still there could be a backward rate constant. That is not the point. The point is that, see, diffusion is occurring, and diffusion is a random process. Okay, diffusion can occur in any direction. So you have to keep the possibility that it can diffuse out of the coated pit. Is that clear? See, that is one of the basic things. The diffusion is a random process, and it can take in every direction. There is nothing that stopping you that it has to be in this direction or that direction, and these are lateral diffusions that can occur. So if there is a concentration or a chemical potential or a number gradient for the receptor out here, in this direction, so there is a possibility that the receptor might move away from the coated pit, though that possibility could be very low, which means that K minus could be much lower than K plus, but still you have to retain that possibility. And then this coated pit, as it is, will form a circle, and we will have to take account the radius of the circle. So the definitions, that the symbol that we are going to use over here is Rp is a receptor in the coated pit, Rn is a receptor on the cell surface, and P is the density of the coated pit or number of coated pit per unit area, and lambda is the rate constant of vesicle formation. Okay. So what we are trying to do over here is that we are again trying to separate out the internalization from the vesicle formation. Okay. So why do I need the density of the coated pit? So the coated pit, density of the coated pit is in terms of number per unit area. Okay. And what are we doing with what kind of balance are we doing over here? We are doing a number balance, remember. Okay. So keeping a track of the number of receptors, we have divided them up into those which are on the cell surface and those which are in the coated pit. And why do we need the density of the coated pit, you tell me? But still, why do we need that?
If we multiply this by area, we can directly, in our model, we can directly convert the number of receptors in the vesicle.
Yeah, or in other words, that is not exactly true, but conceptually it is correct. In other words, more is the amount of, more is the density of the coated pit, then the more is the possibility of the receptor binding to it, right. So if P is the density of the coated pit, and if that density higher is the density, more is the possibility of it binding to it, and that is conceptually correct. The way it will come in our model is slightly different, and you will look at it, what is going on. And lambda is a rate of vesicle formation. Adapted proteins are uniformly distributed in the coated pit and are not limiting to binding. This we already discussed, that proteins are there and the reaction is supposed to be very fast, and this is not what is going to limit. And why are we saying adapted proteins are uniformly distributed? What is the significance of this word? So that we do not discriminate based on where we are on the coated pit. As long as we are on the coated pit, if the adapted protein, the three adapted proteins that are there are uniformly distributed on the coated pit, it does not matter at what geographical location on the coated pit we are. Okay. Is that clear? Okay. Now let us go and look at the model. There is a slight screw up in the arrows over here, but that is not a problem, I think. So as I said, the symbols are Rp is the receptors in the coated pit, Rn is the number of receptors on the cell surface. Okay. Receptor on the cell surface, P is the density of the coated pit or number per unit area, and lambda is the internalization vesicle formation rate constant for vesicle formation. And So you see over here the way we represent it, and what he said is correct, that larger is the number of proteins on the cell surface, on the coated pit, more is a possibility, and the way we include it is making it a second order reaction instead of a first order. Okay. So see, we have to understand this, that this process, why am I talking about this entire process that we are talking about is a conceptual process. In reality, this is not a reaction, it is not a first order reaction or a second order reaction just like that. Okay. So we are conceptualizing a thing which is a diffusion with respect to reaction. So when diffusion occurs, if the concentration or the density of the coated pit in a certain region is more, then the probability of receptors migrating to that particular region is also going to be higher, right. So that is what is incorporated through this second order model. Is that clear? It is not a reaction taking place as it is, but we are conceptualizing this diffusion followed by reaction process by a reaction process, and these rate constants k plus k minus that we have over here are not real rate constants, they are called diffusion limited rate constants, and I will come to that. So the reason they are called diffusion limited, because actual reaction is not limiting as it is written over here, the proteins bind very fast and the whole system is limited by diffusion. So we write it as a second order reaction. So Rn being the free receptors binding with P, the proteins on the coated pits, K plus is a forward rate constant, I mean so called rate constant, K minus is a so called backward rate constant, and Rp to Ri, that you can conceptualize. Can you see that on the screen? Okay. So Rp to Ri is you can conceptualize it as a reaction, and lambda is a rate constant for that. Okay. So ddt of nRn is just minus k plus nRn times Np plus k minus nRn, and ddt of nRp, then this will, both these terms will come in. So the forward reaction formation of nRp and nRp loss due to backward reaction and the internalization process. Okay. So nRt is still, the total number of receptors is still nRp plus nRn. Why is that? Because we still have not considered receptor regulation. We will do that in the let us get done with this model, and then the next part of the model that we do now will consider receptor regulation. So at steady state, nRp, the fraction of receptors in the coated pits is given by nRp over nRt. Okay. So which is because of the constraint, it is nRp over nRp plus nRn, and that is given by from here. Okay. At steady state, if you equate these two equations to the left hand side to 0, you can come up with the relationship between nRp and nRt. That is what we have done over here, and the whole thing can be written in this. So nRp could be written as k plus Np over nRt, and Np is related to the protein density on the cell surface. Okay. So it turns out that from experiments, so this is a fraction that is important and that we can measure, which is the fraction of the total amount of receptors that are on the coated pits. So that number comes out to be around 0.7 experimentally, that is measured to be around 0.7. Np is the concentration of protein concentration. So though no unit of concentration is there, so it's number per, sorry, it's not concentration, it's number per unit area. So 0.31 per micrometer square, micron square, and lambda is the rate constant, so it is 0.25 m in inch inverse. And this is what I think I'd like to attract your attention to, this k plus and k minus you see over here. Is this the first time you're encountering something like this, diffusion limited rate constant, or have you seen this before? No? Okay. So this is based on probabilistic theory. This is obtained from probabilistic. Does this ring a bell? Have you seen something like this ever before? Where? Heat transfer where exactly? No. Not in concentric resistance, but somewhere else. And I taught you that in one of the earliest lectures of heat transfer. I taught you. Whereas this is not a this is not obtained from a, that was a deterministic model we did. This is not obtained from a deterministic model. Heat transfer in gases, do you remember? Not k plus and k minus, but something similar to this. How do you obtain the thermal conductivity in gases? You remember that? We did that in the class. So that had to do, the thermal conductivity in gases had to do with kinetic theory, and these parameters that are coming in between here came in there. So here B, for example, so the K plus, the forward one is given by 2 pi dr over ln B over S, and the backward is given by 2 dr S square over ln B over S. Okay, so if S is the radius of the coated pit is large as compared to B, then backward rate constant is obviously much, as you can see, obviously much smaller than the forward rate constant. Now, what is this? B is half the mean distance between pits, and this, the analogy that you have is in kinetic theory, is the mean free path of the between the molecules. So let's go back to the picture and then you can see. So B 2B is the total distance, mean distance between, mean distance of course, because these are probabilistic models. So 2B is the mean distance between two coated pits, and B is half the distance, and S is the radius of the coated pit. So once the coated pit is, it's not a radius of curvature, but once a coated pit becomes a vesicle, so it is a circular thing and you can get a radius or an effective radius at least. So S is the radius of the coated pit, and as you can see over here, so what is dr? What is dr? Diameter of the cell. No. Diffusivity of the receptor. Yeah. So see, look at these two rate constants, K plus and K minus over here. There is no reaction parameter out there at all. Okay. So that is what I was trying to say, that these are not reaction rate constants any which way, and the reason the reaction parameters do not appear over here is that because reaction is very fast, it's irreversible and very fast, it has nothing to do with the process, and all we are trying to do is this complicated diffusional process we are trying to model using reaction. So you can go and look up a little bit more if you are interested in Brownian motion theory or kinetic theory. So how to do this. So, and it's an interesting area, and if you are interested you can look at this. So we're just going to use these two rate constants, and once we use this, they are here, K plus, K minus and lambda, you can get the fraction of the receptor. Then you can incorporate that into your model, and it's your job as an assignment actually to do that, to incorporate that into a model and show what happens. But I gave you, this is the model, but what I'm asking you is that how do you integrate this into the earlier model, right? We had the earlier model over here, let me go back here. This was a full kinetic model. So if it's a, assume, let's say it's a class 2 receptor. Okay, so how do you, this model for the receptor binding to the coated pit, how do you integrate to this model?
Second equation will replace NLDRL by fraction into NLDRL.
By fraction of F into?
Fraction into NLDRL.
So NLDRL is essentially our nRT that we talked of, right? Is essentially it's not sorry, this is not an LRT. You have to split this up. First you have to split this up into two parts. So an LRT would be NR plus NRN plus NRP plus NLDLR, but I don't think you just replace it by fraction times that, probably not, just a little bit more, because
internalization rate will only be dependent on the fraction, only that part which is associated with the coated bit. So if we replace, this is a fraction of that.
correct, but there is a step of the vesicle formation also, you are skipping that.
Vesicle formation is after internalization.
Yeah, after internalization, but we have to differentiate between these. So what I said is that this is no longer NLDL is no longer then you have to form the NLDLI, then you have to have the NLDLI will have the vesicle formation reaction into it. So just have a look at that, think about how to integrate. So there are two steps that, one is that coated pit attachment to the coated pit, and then the vesicle formation. So how do you integrate that with this model? So that's a task that I give you, and you can do that on your own. Let's move on to the last model that we do in this chapter, which is the regulation, receptor regulation. I try to finish, we don't have a lot of time, but let's try to finish it in today's class. So regulation, receptor regulation during endocytosis. So as I said that receptors are not necessarily receptor number is not necessarily constant. Till now everything that we had assumed that receptor number is not necessarily a constant, but receptor number is regulated. So here what we are saying, in general the assumption that nRT is constant is not valid. Cells regulate, when we talked about this, cells regulate the number of receptor on the cell surface to regulate the rate of endocytosis. So at points of time when there is a immune attack on the system or anything, you probably have a lot of protein, and the body for some reason wants to internalize this protein, it is going to increase the number of receptors that is available for uptake, and as a result accelerate the process. So a kinetic model for receptor regulation should include reversible binding, is a type of reversible binding of LR on the cell surface, internalization by vesicle formation, all this. Sorting, degradation, these are things that we have not talked about. Sorting, degradation, recycle of receptors and ligands, and finally receptor synthesis. So whenever you are talking of receptors, so what have we looked at till now? In this model, what we had looked at was, even if you add this, was up to the internalization step and the coated pit and vesicle formation, but we had not looked at that what happens after internalization, the decrease in the pH we had talked about. But the fact that once the pH is decreased, then it splits up into two parts. One has the ligand, one has the receptor, and the receptors are sent through microtubules back to the cell surface. So when you are going to take a total count on the number of receptors, you obviously have to include all of this, all of these effects, right, which is the recycling of the reactor back to the cell surface, and the final thing is synthesis of reactor. Okay. So, sorry, one more thing, degradation and synthesis. So receptors are being destroyed, receptors are being formed. So three things that are happening and that we haven't looked at and need to look at now is recycling back of the reactor to the cell surface A, B is degradation or destruction of the receptors, and C is synthesis of these reactors. So how do we incorporate these into the model? It's not hard, and let's do that quick. So this is the picture that we need to look at. This is a picture that contains all the effects. So this is a complete model, and what is incomplete, what is missing, can you have a 30-second look at the picture and let me know what is missing in that picture? If I ask you to do a complete model, this is a schematic of general model. General model is fine, but if I want you to make a complete model, is this a complete model or is something missing? Right. The step that we discussed earlier about the binding to the coated pit is missing out here, and if I ask you to do a complete model, you have to add that step in and do the kinetics. It's not easy actually, for not writing the model, but solving this model is not easy actually. So let's go step by step. So this is the case where the receptor ligand binding is occurring on the coated pit. You add the binding of the free receptor to the coated pit, or the ligand complex to the coated pit, depending on the class of the reactor. Add that part over here. Then this is a internalization that is taking place. Now as you see over here that we have these two rate constants here, KEC and KER. Can you intuit what these two rate constants are? Why 2? Why do we have two rate constants, KEC and KER? What is KEC? What does the name suggest? No, it's a complex. C is for the complex, that is the what? Yeah, one for the complex, one for the receptor, but why that?
There will be some free receptors.
There would be some free receptors if it's, if the receptors are already bound to the coated pit, then there could be some free receptors which are there, which are not binding to the complex, but they are on the coated pit. So you have to incorporate them when you go to the internalization process. So the rate of when the internalization of the vesicle formation is there, the rate at which the this is free, this is for free, and this is complex. So the total amount of receptors that are there is not necessarily equal to everything that is there in the complex form. Some are free. Okay. So then this is formed. Now then this vesicle is formed, and then the late endosome and all that stuff, and early endosome, and then what happens between here and here? What process? Acidification happens, and these hydrogen ion pumps open up and pump in hydrogen ion. As a result, the system is acidified, and when the system is acidified, what happens then? Here what I am pointing to, the K-1 will increase, the dissociation rate constant will increase. As a result, the complexes that are there are now going to dissociate, and then they are going to go to two different parts. So these receptors will, as usual, be sticking to the cell surface, and the ligands will be floating around. Okay. Now this ligand that you have, ligand and receptor, both are partially, so this is a general model and it takes into account all kinds of possibilities. So there are two possibilities. One is that the receptors could be recycled back, and there is also a possibility that some of the receptors are degraded. Okay, so you have to account for that possibility. So what we have over here is that K FL is the fraction of these receptors. So if the total number of receptors, say n or something, whatever that is there in Ri, then FL is a FR is a fraction of receptors that are being degraded, okay, that are being lost in the process, because there is always going to be some receptors that are going to be lost. And also you remember a couple of lectures back we talked about the lifetime of receptors. Okay, how many rounds did we say the receptors can do? 150 rounds. 150 rounds the receptor can do, at the end of which it is going to be degraded. So at any point of time there is going to be some receptors who have finished their lifetime, and they are going to retire. So FR is the fraction of receptors which are degraded, and if FR is the fraction of receptors that is degraded, 1 minus FR is the fraction of receptors that are going to be recycled. Now these are then preceded by their rate constants, because fraction is fine, fraction is just a concentration, but the rate at which there is degradation or the rate at which this recycling is occurring is also important. You have to take that into account. Similarly for ligands also, not all the ligands preferably they would like all the ligands to go into the endosome, okay, and be used, but not all the ligands are degraded or are entrapped in the form of lysosomes and used up. Some of them can be recycled back. There is a possibility that some of them can be recycled back. So this is the overall process. The last thing that we need to add is a synthesis. Vs is the synthesis of the reactor that is happening. So at any point of time you can assume that Vs is the amount of receptor that is synthesizing. So I wish I could keep this. Do you want to give it a shot, or shall I? Let's see how much time I think. Shall I just give the model? Because okay, let's go back and go back and forth from the model to the picture. So NRS is the receptor, so let's see, NRS is the receptor on the cell surface. So that's the nomenclature. So the receptor on the cell surface, this is what is minus K1 Cl0 NRS. That is the formation of the complex, right? And this is a backward reaction, K minus 1 NCS. Let's go back again. So K minus 1 NCS, this is a backward reaction, and forward reaction is Cl0 times NRS times K1, fine. So these two terms. Then KER NRS, that's a and that's what is taken up here, okay. Fine. The receptor, the number of receptors that are going into the vesicles, fine. So receptors are formed, and combining with this to form complexes, but some of the receptors, free receptors, are as it is going into the vesicle. So that is this KER times NRS. Vs is the synthesis of the receptor on the cell surface, and the last term KREC FR 1 minus FR times NRI is the recycle that occurs. Let's go back to the picture again. So this is a recycle that's occurring, okay. So NRS, if I am keeping a track on the number of receptors on the cell surface, so what's happening is these two processes, backward and forward reaction, internalization or encapsulation in the vesicle, and recycle back, and of course the synthesis. So this is the total number on the cell surface. Complex is more easier. And by the way, the assumption here is that Cl0, that no ligand depletion, that assumption is there, therefore Cl0 is used here. So complex formation is more straightforward. So K1 Cl0 NRS minus this. So there are two processes, one is formation, that is forward reaction, one backward reaction, and the other one is internalization, just 3, okay. Then what else do you think, what other let's go back to the picture, what other things that we have to take, do a balance on? We have to do a balance on NRI, okay, that is the internalized receptors in the internalized form. So that's what we will do now. NRI is it doesn't have anything to do with, NRI would not have anything to do with what is actually happening on the cell surface. It would have to start from the invagination or the encapsulation process. So NRI is given as KER times NRS times KEC times NCS, okay. minus these two. Why is this added? Why is KER times NRS? Fine, agreed. I think nobody would have a problem with this. Why KEC times NCS? Let's go back to the picture, then you can see. What is NRI? NRI is a receptor, all kinds of receptors that is inside the endosome. Right, yeah. So, what happens here, that the combined receptors that are there with the ligand, once the pH is decreased, the system is acidified, they also give these receptors within the internalized form. So, that is resultant both from the free receptors and the combined, and these two are things that you know. One is these two are the losses that are happening in the internalized receptors. One is because of degradation of the receptor, which is given as K degradation times FR times NRI, and the other thing that is happening is the recycle, which is KREC times 1 minus FR times NRI. Okay. Now, in addition, we the last balance that we are writing over here is NLI, which is the ligand in the internalized form. Why do we have to write the ligand in the internalized form? Because unlike before, where we had assumed that all ligand that are internalized are being used, here that is not the case, because some of it is being degraded and some of it is being recycled back. So, the first part is all right, like we had before, okay, KEC times NCS, but these are terms we did not have before, because we did not take into account the recycling and the degradation process. So, minus K degradation times FL, this is the degradation, and minus REC KREC times 1 minus FL, and both multiplied by NLI. So, this is what we have. Now, if we want to study the dynamics of the process, we simply go and simulate this process. As you can see over here, the solution of this is not as easy as the solutions before, and we have already made this assumption that the ligand depletion does not occur, but still the solution of this is not as simple. The reason being that, you know, first NRI is it's coupled, you know, the entire system is coupled. So, you have NRS over here, NRS over here, NCS, NCS over here, NRI over here, NRI over here. So, all cross terms out there, A, and B is also because you have quantities like Vs which can vary with time, right. So, Vs itself, the synthesis is that is how the regulation process is, you know, affected, because essentially what you do, what does the cell do? It regulates the system by synthesizing more at some point of time or less at some point of time. Okay, you can regulate. What could be the other ways to regulate? You have the set of equations in front of you. What do you think could be other ways for regulating? What are the other parameters? If the cell wants to regulate at a certain point of time, it just decides that it wants to increase the number. So, how you regulate? You have, for example, an immune attack on the system. What would the cell do? It would like to increase what? Which variable? You have 4 variables out here. Which one would it like to increase? No, variable. Vs is not a variable. You have 4 variables out here: NRS, NCS, NRI, and NLI. Which variable would the cell like to increase? NRS, right. It would like to maximize the total number of free receptors on the surface so that they can take up the ligand. So, if it wants to do that, then you see the screen. So, what would be my parameters? So, NRS DDT. So, essentially what it would like to do, it would increase DDT of NRS, right. On the cell surface, it would like to increase the number suddenly. So, if it wants to do that, so that's your clue. So, equation A, what parameters can you tune? You know, it's like a control system. So, what parameters would the body or the cell tune so that you can increase the number of receptors? Fraction is not possible, but yes, you should tune these rate constants, the KREC and the KER, not the KER, sorry, KREC essentially. So, KREC is something that you can tune. You may be able to tune the fraction, but you know, that's hard to tune, because these rate constants are functions of time. The cell can increase the rate constant, cell can decrease the rate constant. Rate constant, again, you know, what do you mean by rate constant? Is it really a rate constant? It's probably not really a rate constant. It's just the speed at which this whole thing, rate constant is the inverse, you know, this would have the inverse, this is the first order rate constant. So, this will have just the inverse of time. So, rate constant is just the inverse of the speed at which this whole recycling process is occurring. So that it can change, it can accelerate the process, as a result of which the rate constant will decrease or increase. Okay, and rate constant will increase if it accelerates the process. If it wants to decelerate the process, then the rate constant will decrease. So that is something easier to control rather than the fraction. So you can do that analysis, but at steady state this is what we do, just a simple steady state analysis. You set all the equations to 0 and you obtain nRT, which is now the total number of receptor, which is nRs plus nc, that is in the complex form, okay, because the complex form takes into account all the things. So, so the free receptors on the surface plus everything that is in the complex form, and this is what it comes out to be. And but yes, of course, the thing that is important here to note is that there is there some meaning in what we are doing, the steady state analysis. Is there some is there some significance to the steady state analysis, that the steady state function, thing that we gave, function that we gave, is there some significance to it? Are we get? Yeah, I mean, so only thing that is, yeah, that's correct to some extent. See, there is not much sense in doing this steady state analysis right here. What we did up to that, the unsteady state analysis makes the maximum sense. Why is that? Because when the cell is regulating the system, there is hardly a steady state. How can you talk about a steady state when the system is regulating? The only thing that the steady state analysis makes sense is that you can measure nRT, right? You can label the ligands and the receptors, and you can measure nRT. So what is the only thing that is important that you can get out here? Not all parameters. There is one particular parameter that you can measure easily, which is VSL, the synthesis rate at steady state. You want to measure what your synthesis rate at steady state is, and that at an unsteady state you can similarly measure the synthesis rate, and then you can compare whether the synthesis rate is increased or decreased. Otherwise, a steady state analysis doesn't cut much ice, because when you're talking of regulation, it's essentially intrinsically an unsteady state process. Okay, so then we are talking about the unsteady state process, and if we are to solve this unsteady state process, these are my initial conditions. You need initial conditions to solve. So at t equals 0, you assume that the number of complexes is 0, nRT naught, which is the initial condition for the reactor, that you can get from this steady state. Okay, is it clear? So what is happening is that steady state is when the system is not requiring itself to regulate. Okay, there is no immune attack, nothing is happening on the system, and the system is quiescent, and then you have the steady state. Then, for example, suddenly an immune attack occurs, and the system wants to regulate, increase your Vs or whatever, increase your KREC or whatever it wants to do. So then for that, your initial condition is going to be a steady state. Is that clear? So this is in a way similar to what we did, this perturbation. We did this in the chemical the chemostat problem. We took the steady state and we perturbed the steady state. So essentially you can think of this as physiological perturbations to the steady state. That is what happens, right, when you get sick. So you have a steady state. When you're normal, when you're doing all right, your body functions at a particular level, some sort of homeostasis is there, and then when you get sick, there's a perturbation from the steady state. So that is exactly what happens. So whenever you get sick or there's an immune attack on the system, this is your steady state in general, normal condition. There's a perturbation, but to evaluate that initial condition from where the system would start, you would need the steady state values. Vs naught is the synthesis at the steady state values, and then you can evaluate the rest. So I think that is all I wanted to talk about in this chapter. I hope that this you want to follow something. There is so if you have any question in a minute or so, we've almost run out of time. But this is the entire process. That's why I put the picture out there. And one of the things we learn from the process is that it depends on the level of complexity you want to go to. You can come up with a very simple model, and then you can increase the level of complexity one step at a time. So is there any question on this? So otherwise we'll conclude this chapter, which is on the receptor-mediated endocytosis. Okay. So if there are no questions, then we'll conclude.